In the last lecture we talked about meiosis, which was the process of getting our genetic material from 2 sets of 23 chromosomes down to just 1 set of 23 chromosomes that can be passed on to the next generation. So in this lecture we're going to start to take a look at some patterns of inheritance. So how those genes on those chromosomes are passed down and how they are then kind of reveal themselves within the organism. So let's start out with an example from nature. So we've got these budgies, or these parakeet birds, which are small birds that can be kept as pets. In fact, I had one when I was little, and I had one that looked exactly like this guy, and we called him Pretty Bird. So that's my Pretty Bird parakeet. Now in the wild, most of these budgies are going to be green and yellow, like my Pretty Bird. But in captivity, we can also find other colors that exist, like blue and white, and a combination of blue and white and sky blue and stuff like that. So let's see how this works. So if we were able to do a little experimenting of ourselves, and we took 2 wild type parents, parakeets, and we mated them. Okay, so in this scenario right here, we mated them, we would get offspring that looked like yellow and green, just like the parents. Okay, however, if, so yeah, there you go, just like the parents. However, if we took this scenario here, where we took 1 wild type, okay, which is the yellow and green, and we crossed it, or we mated it with a sky blue parakeet, again, we would have our first generation of offspring look just like the wild type. Okay, so it would be yellow and green. Now, if we took 2 of these offspring and mated them, okay, what we call the first generation offspring, if we took 2 of those guys and mated them with each other, then we get a whole new thing. We're going to have 3/4 of the second generation be yellow and green like the wild type, and 1/4 the sky blue color. Okay, so we're going to take a look in this lecture and see how this is actually working. Why we get 3/4 looking like the wild type yellow and green, and only 1/4 looking like the sky blue. So to understand this, to understand why we can get, some blue parakeets from, 2 yellow and green ones, okay, we have to venture back to the 1860s and a guy named Gregor Mendel. He was basically the first guy to analyze this pattern of inheritance in a systematic, scientific way. Other people had noticed these patterns, but they never really kind of bothered to do tests and run their own crosses and stuff like that. So Gregor Mendel, he was a German monk back in the 1860s, had a liking for pea plants. So he had a garden full of pea plants. So he noticed, as he was fiddling with these peas, that the parents are going to pass on to offspring these discrete heritable factors that are responsible for herited traits. Okay, so he didn't know anything about DNA or genes or anything like that back in the day. So he called these, what he called heritable factors, we now call genes. Okay, and he noticed that they retain their individuality generation after generation, whether they're expressed or not. They're still going to be there somewhere in the organism. So a little background on peas will help us to understand this process a little more. Peas are very easy to grow, and they have many readily distinguishable varieties. Right, we can have pea plants with purple flowers and pea plants with white flowers, or pea plants with green seeds versus yellow seeds. So they have very distinguishable varieties. And they could exercise complete control over breeding them, because inside of the flower we have the male and female parts here, the stamen and the carpel. And so the female part is the carpel right here, and the male part is the stamen. Okay, these little guys that make the pollen. And basically in an enclosed flower, they are going to self-fertilize. So whatever pollen is made by the stamen, okay, is going to kind of be directly, the carpel here is sticky, so it's going to stick directly, to the pollen, and it's going to self-fertilize. Okay, so that's usually how peas work. However, Mendel figured, all right, I can mess with nature a little bit, and I can cross different varieties of pea plants by fertilizing them by hand. So he would remove the stamen and transfer the pollen from one pea plant to another using a paintbrush. Okay, so he would remove the pollen from inside one of the pea plants and take it and fertilize another pea plant with it. Okay, so in our true breeding pea plants that self-fertilize, what he noticed is that they always give the same offspring, like the identical offspring to the parents. So the purple ones, if they're able to self-fertilize, will always have purple flowered offspring. And the white ones, if they were left to self-fertilize, will always have white colored offspring. Okay, so he did notice that. So what he wanted to do was cross-fertilize them and see what happens. So to set up what he was going to do with his cross-fertilization, to understand that, we need to know kind of some vocabulary here. So when we cross two true breeding varieties of peas, okay, we're going to get what's offspring, what we call the F1 generation. Okay, so that's going to be our offspring. So this is basically our F1 generation here. Here's our F1 generation here. Okay, the true breeding varieties, those are parent generation, or P1. So these guys would be P1s, and these guys would be P1s. Okay. Now, if we breed two of these F1s together, we're going to get what's called an F2 generation. Okay, and so that's what these guys are, is our F2 generations. Okay, so when we talk about, peas, oops, this should say 1. Yikes. When we talk about peas or F1s or F2s, you'll kind of have an idea as to where it goes along in the order of genetics. It's also important to point out that peas are going to have many traits that actually occur in pairs. So you know how in humans, like, we've got brown hair and black hair and blonde hair and red hair and combinations of those, and dirty blondes and towheads versus regular blondes, stuff like that. In peas, we just have purple flowers or white flowers. Okay, we don't get stripes, we don't get polka dots, we don't get some other type of flower. Same thing with flower position. It's either axial, okay, which comes off from the sides of the stems, or terminal, which comes off the ends of the stems. Same with seed color. We either have yellow seeds or green seeds. We don't get polka dots, we don't get stripes, we don't get blue. And then seed shape either comes as round or wrinkled. Again, not a combination, and not anything else than round or wrinkled. Same thing with the pod shape. Okay, we can either have inflated or constricted. The color could either be green or yellow. They could either be tall stems or dwarf stems. So they all come in two alternate forms. Okay, all of these P traits come in two alternate forms, which is actually very useful for when we're trying to figure out kind of how genetics works and how inheritance of genetic material works. Okay, so we're going to investigate, just like Mendel investigated, one trait at a time, and we call that a monohybrid, because mono means one, right? So our parent plants are going to, in these crosses, we're just going to look at one different trait at a time. So purple flowers versus white flowers. We call that a monohybrid cross. So when he mated a purple flower with a white flower, he got an F1 generation. All right, so these are the parents. We got an F1 generation of all purple flowers. Okay, nothing but purple flowers right here. Now, if we mated two of these F1s together, then we got 3 purple flowers and 1 white flower. So the white, the white flower genetic material was there, and it came back in the second generation. Okay, so this is what we call our F2s. So it never disappeared, although the genetic material never disappeared, but in our F1s, it sure seemed like they disappeared because all we had was purple flowers. But when we mated the two F1s together, we got our white flower back. So that's why, basically Mendel said that we have these heritable characteristics that retain their individuality generation after generation and can come back in characteristic at one time or another. Okay, so we can see that 2 factors are going to exist for flower color. We either have purple flowers or we have white flowers, so it has 2 alternate forms. So the heritable trait is the flower color, and the 2 forms, or the 2 alternate forms of that, are either purple or white. All right, so these genes that are, even if they're not displayed, they're still present, they're still seen, because we can get that white flower, remember, back in our F2 generation. So for each inherited trait, an organism is going to have 2 genes, one from each parent, right? And so in these genes, we have different alternative forms, or what we call alleles. All right, so this is important to kind of know. And you can either be the same allele for the genes or different alleles for the genes. So let me kind of explain a little further. So for each characteristic that we have, it is controlled by a gene. So hair color is controlled by different genes. Now these genes can have 2 different forms. We could either, like in the pea plant, for example, we have genes for flower color, and we have 2 alternative forms of that gene. We can either have a white flower gene or a purple flower gene. Okay, so what we call those 2 alternative forms is an allele. So we can say we have a white allele or a purple allele for the flower color gene. Alright, so sperm cells and egg cells are going to carry only 1 allele for the trait, since they're separated during meiosis. And when we get fertilization, those 2 alleles will come together to give the resulting offspring 2 alleles for that trait. And it'll just depend on which one is dominant over the other, or whether they're co-dominant with each other, to see what actually the organism is going to look like. So when 2 genes of an allele are paired up and they're different, and one is expressed and the other one has no effect. So for example, in our F1 generation here, remember we got a purple allele from one parent and a white allele from another. So the F1 generation is going to have 1 allele of each, except only the purple one is expressed. Only the purple one shows through. So if one is expressed and one isn't, we call that dominant versus recessive alleles. And the dominant alleles, we're going to use an uppercase letter when we do these Punnett squares. So we can say, for color, we can say big P for purple, okay, which is the dominant one, and little p for white, which would be the recessive one. So a couple more vocab words. We've got phenotype, which is the expressed physical traits. So that would be in our F1 generation here, that would be the purple flower, versus the genotype, which is actually the genetic information. So even though the F1 has a phenotype of purple, it still has a genotype of big P, little p. Okay, like those don't look very different, but there's a big P and a little p, one coming from the purple flower, which is dominant, and one coming from the white flower, which is recessive. So for all the 7 traits that we can study in pea plants, it looked, to Mendel that the, one parental trait disappeared, okay, whether it was the white flowers or the green seeds or whatever, seemed to disappear in the F1 generation, but then reappear in the F2 generation, because remember, those alleles are still there. They're passed on generation to generation. It'll just depend on how they are expressed. Let's now link what Mendel saw in the pea plants to what we know about DNA and genes. So an organism is going to have, remember, 2 sets of homologous chromosomes. One set from mom, okay, so I've always mentioned that we'll do mom as red, and one set from dad. Okay, so the dad will be blue. Now, on these genes, which are made up of DNA, we have different locations, or special locations where these alleles, or different forms of the gene, exist. So if we take a look here, we've got different locations on the homologous chromosomes where different alleles are going to sit. If they are identical alleles, meaning that from mom you get a big P and from dad you get a big P, okay, we call that homozygous. Okay, so homozygous can be dominant, like big P, or it could also be recessive, like here, little a, little a. Okay, so it can be dominant or recessive. Now, if we have 2 different alleles for the same gene, we call that heterozygous, right? So if we have a big B from mom but a little b from dad, we call that heterozygous. Okay, so true breeding organisms, okay, pure breeds as we call them, are always going to be homozygous. They're going to have just one type of allele for each gene. Okay, so let's talk about a principle of independent assortment. So now we're going to expand our thinking. Instead of just looking at flower color, we're going to look at 2 different traits at the same time. When we do that, it's not called a monohybrid cross anymore, because remember, mono meant 1 trait. It's called a dihybrid cross. So dihybrid crosses, we look at 2 traits at the same time. So when Mendel crossed plants producing dominant round and yellow, okay, so there's our 2 different traits, yellow and round seeds, okay, with plants producing a recessive wrinkled and green. So again, we've got the green and the wrinkled seeds. So those are the 2 different genes that we're going to take a look at. Seed color and seed shape. We can either have dominant yellow for color or recessive green for color. And again, the 2 alleles for seed shape would be dominant round and recessive wrinkled. Okay, so again, if our parent plant, our P1 generation, is yellow and round, okay, mated with a green and wrinkled, remember those gametes are, will have single combinations of these alleles. So what he noticed is that in the F1 generation, okay, this guy right here, they were all yellow and round. Okay, that was the phenotype, all yellow and round, and they would all have the same genetic combination of, big R, little r for shape, and big Y, little y for color, and big Y, little y for the shape. Okay, so yellow being the dominant color, round being the dominant shape. Now, if we took 2 of these F1 generations and we mated them, then we get this big bang down here of an entirely different, pattern that we've seen before. So we have 9 different genotypes, okay, so 9 different genetic combinations of the 2 alleles, and 4 different phenotypes. So we can see here, okay, that we ended up with some yellow and round ones. We ended up with some green and round ones. We ended up with some seeds that were yellow and wrinkled, and some seeds that were green and wrinkled. Okay, so we have a whole bunch of different combinations when we take a look at 2 different traits at the same time. Because we can see this wide variety of phenotypes and even bigger variety of genotypes in the F2 generation, we could see that each pair of these alleles is going to separate independently of the other pairs of alleles during gamete formation. So it's not that every time you have a yellow seed it has to be round because those alleles go together. Okay, that's not it at all. That's what this principle of independent assortment means. Basically, it means that the genes, the different alleles for the genes of seed color versus seed shape are going to segregate independently. So we didn't see here that every time it was yellow it had to be round and every time it was green it had to be wrinkled. We got different combinations. So that means that these alleles can separate or segregate independently of each other when meiosis is occurring. Alright, so that's where we're going to conclude this lecture on. We are going to have one more lecture on more patterns of inheritance and take a look at some more details of human genetics rather than in the pea plants. So we'll get a little more of a personal perspective on genetic inheritance.